So we are going to proceed with the characterization technique, and today in lecture 6 we are going to talk more about the X-ray diffractometer. In the last lecture 5, please refer to that, that was the part 1. We have given a brief overview of the instrument. Today we are going to look into more detail of the instrument, okay, the X-ray diffractometer with some images, and when we will be starting off with our physical classes and sessions, we will be looking into the instrument in more detail and understanding it from a very close point of view. Okay, so if you look into the goniometer, which I have told you in the last class, okay, we are talking about parts of diffractometer now, okay, and that actually mostly lies in the goniometer setup itself. This is a typical scheme of the Bragg-Brentano type diffractometer. So if you remember in the last class, I told you there are two configurations, which is theta to theta configuration, and we have the theta theta configuration. Okay, so it depends on what kind of instrument you are using, whether the source is fixed or not. Depending on that, we have got the different configurations. So you can refer to that in the last class lecture again. But right now what we are going to see is the different parts of the diffractometer, and we are going to go from left to right. Okay, so what we are going to see first is the X-ray tube. What are the parts of an X-ray tube, how it is generated? We already know it partially, but still we are going to look more into it. Then we're going to look into something which is called as the primary optics. So primary optics is on the receiving end, okay, receiving end. So that means from where the X-ray actually falls on the sample, that end I'm talking about. So we have the solar slit. We are going to know about something which is called the divergent slit. So we should know these terms. Okay, right now just get acquainted with the term. Slowly as we proceed, we are going to look into what are the uses of different parts of this X-ray diffractometer. And then we are going to look into the sample and the sample state. This is a very small part. And then what we are going to do is we are going to look into the secondary optics. So this side is nothing but the secondary optics. So in there we have got something which is called the anti-scatterer slit. We also have got something which is called solar slit, and then we have got the receiving slit. Okay, and then finally the detector, how we are going to detect it. Detector we have already studied. If you remember scintillation detector, we have done it in our class. So that same detector we are going to use even in an XRD machine also. Okay, and then please remember or note this word solar slit. It is not SOLAR as we are usually acquainted with. It's SOLLER. Okay, SOLLER. So please remember this, and then let's start our journey for this. And then let's talk about the source first. So we already studied partially about the source, but we will just give a brief here. The source, what it does, it produces just the X-rays. Okay, and this is the most important part of an X-ray instrument, because in an X-ray diffractometer we use these X-rays to analyze the sample. Okay, and then we have learnt a lot about this already. Now typically the source is an X-ray tube. Now X-ray tube is not a simple setup. It has got a too much of electronics involved. Okay, but it's an X-ray tube which generates the X-ray, and it's basically an evacuated ceramic or glass vessel. Okay, that basically contains the anode and the cathode. So I hope all of you remember anode cathode concept. So what happens is that contains a tungsten filament as cathode. So the tungsten filament is a cathode, and this tungsten filament will emit electron. Okay, and then we have got an anode here onto which these electrons are accelerated with a potential of several 10,000 of volts. So if the voltage is very high, these electrons which are generated by the tungsten filament, called the cathode, we will go and hit the anode, okay, at a very high speed. So if it hits at very high speed, what happens is from the anode some electrons are extracted, as a result of which the electrons from the outer shell will fill the inner shell electron, and finally we will be getting the X-ray. Okay, so this is exactly what happens. I think we have already talked about it. So several process leads to this. Now that means after the fast electron hits the anode, which is copper mostly, but we use other sources also like cobalt and molybdenum and so on. So several process leads to the emission of electromagnetic radiation in the X-ray as the electron hits the anode target. This process we have already discussed in our last lecture. Please refer to that. So one of the processes, electrons are quickly decelerated by passing the nuclear core. So electrons are charged particles which are negative, and the nucleus is positive. So as the electron passes nearby the nucleus, it is decelerated, that means the speed comes down, and then from there we get an effect which is called the Bremsstrahlung or the white radiation effect. Okay, this is one of the process. The second process is the accelerated electron will hit an inertial electron. We have already studied about that. And then what happens from the target material, so that target material will be copper, cobalt and so on, and it will remove them. And then what happens, it leaves holes behind. So if there are holes, that holes needs to be filled. Okay, so that can be filled by an outer shell electron, and then in the process we will be generating K alpha line and K beta lines. Now the K alpha line can be K alpha 1 or K alpha 2, and K beta lines are also generated, which are the M lines. Okay, if you remember in the last class we have discussed, and these are nothing but unwanted byproducts. So when we are actually going to study a sample, we really don't need a K beta line, and that is where we use the filter. So please refer to last class and this class together so that you clearly understand what are K alpha and K beta line. Okay, that is lecture 5. So can we always use copper as a source? The answer might be no, because sometimes depending on what kind of sample we have, we might also be using other sources. Okay, but copper no doubt is the most commonly used anode material in the powder XRD. Now if a sample shows fluorescence, I hope you know fluorescence, okay, because what happens is sometimes some samples, if there is a matching of the energy level difference with that of the incoming radiation, there can be the process of fluorescence which will actually interfere with the signal that I am interested in. Okay, so it will show a fluorescence effect, then the sample will simply absorb and radiate X-rays from the copper anode. So what will happen, it will absorb and re-radiate X-rays from the copper anode. So what happens, the copper anode gives out X-ray that is getting absorbed and then re-radiated. Okay, and this will actually lead to a large amount of noise in the output. I don't need that. Okay, so I might have a signal something like this, and then I might have something like this, and these are nothing but noise, but I cannot differentiate it with the real signal, and as a result this data is quite useless for me. Okay, so based on this I have to select my source. So if the sample is fluorescent in nature, I usually don't select copper. So what do? I can use some other source. For example, let's say I will use a cobalt anode. Okay. Now this cobalt anode, what it does, it provides less signal intensity, but will also remove the noise caused by the fluorescent background. So if we have a fluorescent background, okay, which is possible because the material is fluorescent, this will have a less signal intensity, as a result of which the process of fluorescence might not actually happen. Okay. So because of this, we can actually use a different source. So please remember this slide. Okay, and if I am asking you a question, when can we use a cobalt source in place of copper source, give one example, you can explain this. Okay, and probably this will be a 2 marks question. So next let's go to the primary optics. Now what does this primary optic do? It usually controls the incoming signal that we have, that this means the incoming X-ray. So what happens in the X-ray tube, we have generated the X-ray and then it is fed into the primary optics. Now this primary optics will control the beam produced by X-ray source and manipulate it into forms more useful for diffraction experiments. Okay, so we are going to basically focus it in a better way. Okay, so I think this image I should put it later. Okay, so the first thing that we need to show is the solar slits. Okay, as I mentioned, please try to remember this term. Okay, I'll just probably go back here. Okay, so we will look into the solar slit first. Okay, we are going to look into the solar slit first. Now what happens in the solar slit, it will reduce the axial divergence of the X-ray beam to less than 6 degree, in some instrument even less than 4 degree or 2 degree. Basically we want to have a peak which is symmetric in nature, and for that we want to reduce the divergence in the axial form, as well as we also want to reduce the divergence in the height. Okay, so that means the solar slit will reduce the divergence in the axial side, okay, in this direction, and this reducing the axial divergence will reduce the peak asymmetry in the output. So if you have got something like this, okay, this is a symmetric peak, but if you got something like this is more of asymmetric in nature. Okay, it's not the sharpness. Okay, please remember this. Okay, I think I've drawn the figure wrong, it's not very correct. So it's something like this. Okay, So this is more asymmetric, this is more symmetric in nature. So we need a symmetric peak, and then we use the solar slit for that. Okay, what does the divergence slit do? A divergence slit, which is actually after the solar slit, reduces the height divergence. So you can also have a divergence along the height. Okay, so we want to reduce that also. That means we want to reduce the beam spread. This narrows the peak and increases the resolution of the output. So what is this actually is something like this. So if you look into this image, we have got the axial and the beam divergence here. So this is your axial divergence. So this number 1 is your axial divergence, this direction, and if you look into the height, this is your height. So this is your height divergence. So basically we use a divergence slit and a solar slit to minimize both these kinds of divergences. Okay, next we have got something which is called monochromators. I think this term you have already heard before. Monochromatic light source means it has got a single wavelength. Now if you have a k alpha, k beta, k alpha 1, k alpha 2 lines, that means they are not really monochromatic in nature, because k beta and k alpha will have two different wavelengths. So what I want to do is I want to filter out the k beta radiation and the white radiation, okay, or the white radiation, and for doing that we use something which is called as the monochromator. You have to remember that k alpha 1 and k beta alpha 2 line are very close to each other. The wavelength difference is very narrow, so they can be considered as almost to be monochromatic. In practical application, actually it is the average of k alpha 1 and k alpha 2 that is taken. For example, if we consider 1.54 for copper, that is nothing but the average of these two lines. Okay, now you have to understand this. I think you have already studied about sodium lamp as using a monochromatic source for studying certain phenomena like Newton's ring in lab and so on. Okay, they are not perfectly monochromatic, but their wavelengths are very near to each other, 5890 and 5896. So we take the average of 5893. Similarly here also k alpha 1 and k alpha 2 are very close to each other, hence they can be considered to be almost monochromatic in nature, the source. So you have to remember this. If we consider a high resolution diffractometer, it will use a single crystal monochromator after the X-ray source. Only monochromatic k alpha 1 radiation is allowed to pass through. So if you are going to use a very high resolution diffractometer, that means you don't even want k alpha 2 line, you want only k alpha 1 line. Okay, for that what we do, you can use a single crystal monochromator. It will only filter out k alpha 1, so this will be purely monochromatic in nature. So as I told about the asymmetry part, this is the asymmetry part. So if you don't have any solar slit connected, there might be a divergence of, let's say, 10 degree and so on. On this is something which it will look like this. Okay, now if you have a 6 degree solar slit, that means it is going to minimize the divergence within 6 degree, and if it is 2 degree, it is going to minimize the divergence 2 degree. So see, this is a very important part, the solar slit. Now if it minimizes the asymmetry up to 2 degrees, that means you are going to see a kind of peak shift. So that means originally the peak should be here, but because of this solar slit, not using a solar slit, rather the peak was here. Okay, so there might be a slight change in result, you might not be getting a perfect answer. So that means we need the solar slit, which forms a very important part of the X-ray instrument. So this was the primary optics. Next we have got the sample holder and the stage. Now this is a very simple thing. Okay, you have a sample holder where you can put the sample. There are different kind of stages or different kinds of sample holders that are used. So most of the diffractometers will actually come equipped with several kinds of sample holders for different types of specimens. Okay, because in an XRD instrument you can study powder samples, bulk samples, thin films and so on. But if you want to study a powder sample, you have to use a different sample holder compared to a thin film sample. Okay, so it depends on what kind of instrument you are purchasing. You have to purchase additional sample holders or not. It all depends on the specifications that you need for your setup, okay, or for your sample. So in the near future, if you are doing any kind of experiment, you can actually study more of powder, and if you study powder sample, we already have some sample holders in the lab which you can definitely see later. Okay, so the best results are basically obtained with rotating sample holders. So it is also important that the sample holders should rotate. Okay, so it is fixed onto the stage and the stage rotates, the sample also rotates. Okay, because of which we are actually getting the theta 2 theta configuration and so on, and this actually improves the measurement statistics, because what happens is the X-ray is getting incident on each and every part of the sample. Okay, however, the most severe error that might arise is the sample preparation. So you have to prepare the sample very carefully. You have to, if you are using a powdered sample, you have to crush it very fine. if you are studying a thin film, it should be well distributed within the layer that or within the substrate that you are putting a sample on. Okay, now you can actually use this idea, and both result in a significant shift or peak position can make the interpretation difficult. So if your sample is not prepared or properly filled into the sample holder, there will be a peak shift. You are going to get a different result altogether. You are expecting some material, you see that there is too much of shift, it might not be even accepted in relevant journals. Okay, so that's very important. So next we have got the sample stage, but before that we'll just have a look at the sample holder. So these are the different kinds of sample holder that we have. Okay, so it depends. So see, if you are actually putting up some powdered sample, you make sure that this portion which we have here, this portion, yellow portion, okay, this should be totally filled up and it should be leveled off. Okay, so that's important. So how do you fill the sample, that is also important. You should not have larger grains, it should be all powdered form and so on. Okay, or you can use these kind of sample holders also. This is also used in our lab. Okay, so you can check that. Next we have got the sample stage. Now what is sample stage? That's quite simple. The sample stage actually keeps the specimen fixed in the right position during the experiment. So obviously you are going to put your sample holder or fix your sample holder into the sample stage for the calculation. So the sample holder is mounted on the sample stage, which keeps the sample aligned in the beam and controls the movement of the sample. So that also forms a very important part, because ultimately you are going to put the sample there. So a sample stage will look something like this. Okay, and you are going to put the sample somewhere here. Okay, so we can have a closer look when we actually look into the real instrument. Okay, so next we have got something which we call as the secondary optics. Now the secondary optics is on the other side of the sample holder or the specimen So the secondary optics will retrieve the diffracted X-rays from the sample. So what does your primary optics do? The primary optics receives the X-ray from the X-ray tube, and after it crosses the primary optics, it gets incident onto the sample, which generates X-ray again. Okay, the diffracted X-ray rather, and that diffracted X-ray is received by the secondary optics. Okay, so that's very important here. So the components of the secondary optics are slit. We have already seen it in the first image. That is called an anti-scatterer slit. Okay, please don't mix up the term. In exam, if I ask you what are the component of a secondary optics, you please don't write down just a slit. Okay, you have to write down the specific name. You have to write down anti-scatterer slit, solar slit, receiving slit, because each of that has got its own specific application. So you cannot just generalize by writing it as a slit. Okay, you already know what is a slit. Okay, I hope you remember from your lower class ideas that what is the difference between slit and grating. So basically when we say slit, a slit is a very narrow opening, and if you have got a number of slits which are placed parallel to each other, these are basically called what? These are called, hope you remember this, grating, right? So this is called grating. So we are not using any gratings here, we are using slits here. Okay, so the receiving slit which we have is either directly followed by a detector. So you can get the signal incident on the detector immediately after it crosses the receiving slit, or it might be again followed by a secondary monochromator to again filter out the unwanted signals. Okay, and that secondary monochromator can again be a single crystal monochromator. Please remember this is the beauty of giving online lectures, at least one of the benefits. And if you don't understand a point, you can actually go back to the slide, listen to the lecture again and try to understand it. Okay, so if you don't understand, I am being too fast, you can definitely slow it down and try to understand it. Okay, we need to cover a lot of things, so I am going a bit fast here. So next we have, let's try to understand each role of each of these slits on the receiving optics, okay, or the secondary optics. So first we have got the anti-scatterer slit. So it's quite obvious, the X-ray which is coming out from the sample might get scattered. Okay? We don't want that. So what happens is it will reduce the height divergence. Okay, it is something which is relevant to something which is done at the divergence slit at the other part of the optics, so the primary optics. Okay, it reduces diffusely scattering X-rays which are due to the amorphous or air scattering. So So there might be some scattering because of the air or the amorphous materials which are present within your crystalline material. Okay, it actually reduces that signal, and this obviously results in the reduction of the noise. So final result is important that there should not be any noise. So what is noise? It means that if I get a signal something like this, which is an XRD peak, I don't want too much of noise here. Okay, this is something which I don't want. Okay, so if I have a noise, might be there will be a peak which is very much similar to this, but this will actually be a noise which I don't want. So this can be reduced by using these kind of slits. Okay, so next we have got the solar slits here. Okay, so the solar slit is reducing the axial divergence. You can refer back to the previous slides where I have talked about solar slits. Next we have got the receiving slit. Okay, so the receiving slit, what happens here, that reduces the X-ray beam height divergence. So it has already been reduced. If there are further divergence, it is again reduced. Okay, so what we want is, we want a very fine signal that should enter the detector. So it again removes the diffuse scattering from the previous optical element. What was my previous optical element? My previous optical element was solar slit. So if solar slit has missed out on filtering out some of the signals, okay, which I don't want, unwanted signals, that can be actually achieved by something which is called the receiving slit. Okay, this receiving slit will improve the instrument resolution by removing the scattered radiation. Okay, so the resolution will be even better. So the secondary monochromator, what it does now, next we have got the secondary monochromator. It might be attached to your instrument, it might not be attached to your instrument, depends on what kind of instrument you are using. Okay, but if we use a secondary monochromator, it will actually suppress the K beta radiation and the white radiation, which are actually again might be present, okay, because of the scattered radiation, or even if it is missed out in the primary optics. So it will only allow the K alpha doublet to pass through the detector. So the aim is to get into the detector, the X-ray, the diffracted X-ray, and that diffracted X-ray should only be the K alpha line. So if we have the detector here, the signal that we should get is also only K alpha. So if we can use some kind of slit, okay, or the secondary monochromator, and if we initially have, let's say, K beta and K alpha, we have got K beta and K alpha. Okay, after crossing this, only the K alpha line will pass through and the K beta will be again filtered out. Okay, that is the role of secondary monochromator. Then that is again very important. Okay, so that means the advantage is basically to remove the fluorescent radiation originating from the sample. So if you have got any kind of fluorescent radiation that is present in the sample, that should be removed, because my aim is not to study the fluorescent radiation, my aim is to study the crystal structure and so on. So detect the X-ray and put it into the detector. So monochromator types, there are different monochromator types and the relative intensity of the characteristic line. So you have studied so many monochromators here. We have talked about the filters. Okay, we have talked about the secondary single crystal and the primary single crystal. Okay, so if we just use the source, we don't use any monochromator there. Mostly the K alpha line will be having an intensity of 100%. The K alpha 2 line will be having an intensity of 50%. Okay, and the K beta line will be having an alpha intensity of 15 to 30%. So these are relative intensities. If the K alpha line is 1, okay, and then if this much is the height of the K alpha 1 line, this much would be the height of K beta, 50%. Okay, I hope I am doing it correctly, the scale. Okay, and then K beta would be this, and K alpha 2 would be this, K alpha 1 would be this. So K alpha is maximum intense. Okay, so if you use a single crystal monochromator, you are going to get only this. So imagine you are going to get a very good signal out of it. Okay, it's going to be purely monochromatic in nature. But on the other hand, if you are not using a single crystal monochromator, you are going to get a mixture of these two. Okay, so that means the intensity might come down. Okay, but we don't need K beta, so this is something which is not needed. So that's why we use a filter. So if we use some normal filter, okay, solar slit, divergence slits and so on, okay, even we use this, then what happens, the K alpha and K beta, they are alpha 1 and K alpha 2, they are not filtered out, K beta is filtered out. But if K beta is filtered out, it's not that it will be totally filtered out, some of the signal might still be left out, okay, 1 to 3%. So this is where I talked about the previous slide, where I was talking about this secondary monochromator suppresses the K beta radiation. So if we use it on the secondary optics, this 1 to 3% will also be filtered out. Okay, you have to understand this, very much logical. And so we have got the 1 to 3% part. Okay, but if we use this secondary single crystal, what happens is we have K alpha 1, K alpha 2, we are very happy with that, we are going to get a signal, and K beta we don't need, it is going to be totally filtered out. So this is actually optional. Okay, so I just want to tell you that this is an optional option. Okay, This is optional. It might be there, it might not be there, it depends on what kind of instrument you are using. Okay, it definitely takes the cost higher. And if we use a primary single crystal, not a secondary, we are using a single crystal which is primary in nature, not a secondary single crystal, then it will also filter out the K alpha 2 line and K beta line, which will be only left out with K alpha line, which is best for our results. Okay, so that is what is important here. Okay. Next, we are just going to look into the detector, which is the final part. Okay, now the detector, what happens, it will actually take the intake, the X-rays which are getting diffracted from the sample will be the intake to the detector. That means that X-ray will be actually detected by the detector. Now, one of the most widely used detector is scintillation counter due to its sturdiness and reliability. Please study again scintillation counter. We have already done this in this semester. I have also uploaded the lecture on YouTube. You can check it. Okay, so that you can again revise this. Okay, this is how our detector works. It will basically have a photomultiplier tube which will actually improve the signal and so on. Okay, so what happens is it consists of a scintillator, a material which is basically produces flashes of light. It will basically a material that emits visible light on exposure to X-rays, and these fluorescent radiation causes a photocathode to emit photoelectron. This radiation will emit a photoelectron which will be amplified by a photoelectron multiplier. A photoelectron multiplier will have something which is called the diodes and all. Okay, I hope you remember these terms. Please refer, this is out of the scope of this particular presentation. Okay, and then this scintillation detector along with the photomultiplier tube is connected to the electronic equipment, and this will actually amplify the signal to be read by the device. Okay, and from this we are going to get a result, and then we are going to analyze it. Okay, so this is what we are going to get here. So finally you are going to get, this is something which is going to get in the computer screen, and this is what we are going to analyze. So I hope this is clear. Okay, we have gone into the instrumentation part. After that we have the result, and we are going to analyze the result and what is the output for that.